Hello and welcome to this Introduction to Microbiology video. I'm Dr. Alid Roberts, and in this video we're going to look at the fate of host cells following viral infection and the knock-on effect viral infection has. And the reason why we need to concern ourselves with the fate of host cells is because different viruses have different effects. Now, all viruses hijack and replicate within a host cell, producing progeny virions using the host cell machinery. However, different viruses have different biology, resulting in different interactions, and because of this, there are a number of potential fates for the unsuspecting infected host cell. The first of which, as we've discussed in previous videos, is where the virus leaves the host cell, which can be either in a destructive or a non-destructive way. Now, when we talk about the destructive release of progeny virions, it is usually associated with the process of cell lysis, whereby the release of virions is the result of the cell bursting open uncontrollably. Then on the flip side, certain viruses can remain within the infected host cell, and in order to achieve this, the cell must remain somewhat intact and functional. Now, when a virus remains within an infected host cell, it can lead to one of three different cell fates. The first is a period of latency, whereby the virus remains hidden within the host for a period of time before it is released via the cell lysis pathway. The second is a period of persistence, whereby small amounts of the virus are released from the host cell without compromising the cell itself, and so the host cell will remain living. And finally, the final cell fate is that of transformation, where the viral genome is incorporated into the host cell genome so that when the cell replicates, so does the viral genome. So what we're going to do now is quickly go through each of these different cell fates and see what exactly is going on, starting with cell lysis. So as I said earlier, this is where our progeny virions are produced within the host cell and released in a destructive manner so that they can go on and infect nearby susceptible cells or be transmitted to a new susceptible host. Now, as a result of this lytic process, host cell death is unfortunately unavoidable, as the release of progeny virions causes irreparable damage to the cell membrane, meaning the host cell is no longer able to regulate an internal environment as cell contents start to leak out into the external environment. Now, most viruses will exert some form of lytic effects on host cells at some point in time. When, however, all depends on other aspects of the viral replication process and its overall biology. Now, moving on, our second cell fate is that of latency. This is where the virus infects a susceptible host cell, but it does not undergo active replication within the host. Therefore, the host cell does not produce viral particles, and the virus remains in somewhat of a dormant state. And if we were to look at this on a graph, we would see that during an acute infection, there would be a spike in virions within a host as they replicate, reducing as our immune system starts to kick in. This would then be followed by a period of months or even years, whereby the number of virions within the host remains relatively low, below a threshold number of virions required to initiate reinfection. Now, what this means is that because virions are always present within the host cell, there is a possibility that reinfection may occur at some point if the number of virions goes above this threshold level. And so if this was to happen, we would refer to the reactivation event as a latent infection, and this is where the progeny virions have almost switched from a dormant state back into an active state, readily undergoing viral replication and spreading throughout host cells. Now, one of the most common infections associated with latency and latent infections is that of chickenpox. This is a childhood viral infection caused by the varicella zoster virus, which can undergo a period of latency, and when it does, this varicella zoster virus remains in an inactive form within the dorsal root ganglion nerves. Then, at some point later on in life, the virus can become reactivated, resulting in the adult infection known as shingles. Now, this reactivation process usually follows a period of lowered immunity within the host, allowing the varicella zoster virus to replicate in sufficient numbers to break through the threshold number of virions required to initiate that reinfection. Now, unlike chickenpox, which affects the entire body, shingles is usually limited to a single bundle of nerves, resulting in this very condensed area, usually on the upper body, being affected. Okay, so now we're going to move on to our third cell fate, which is where the virus forms a persistent infection. Essentially, progeny virions are released from the host cell continually at a rate that does not kill the host cell, but it allows the virus to remain present within the host almost indefinitely. Now, this is actually a common feature of enveloped viruses, and as we can see from this graph, we have our acute infection, which is where the number of virions within the host peaks, resulting in visible disease. However, the number of virions present after this is at a level that stimulates an immune response. Now, a good example of a persistent viral infection is that of Ebola, which is caused by the Ebola virus, which results in hemorrhagic fever. Now, it's not necessarily known as being a persistent infection, as many associate Ebola as being a very deadly disease that can quickly kill someone. However, it can form persistent infections in survivors, and that is because the virus can be found in ocular fluid. Now, the reason it is found here is because it is what we call an immune privileged tissue, meaning the immune response does not scan this area, as any inflammation could prove problematic. Therefore, the presence of the Ebola virus goes unchecked. Now, unfortunately, or fortunately, depending on how you look at it, whilst the presence of Ebola virions within the ocular fluid doesn't result in reinfection, it can cause secondary problems, such as cataracts and various other ailments of the eye. Okay, so now we have made our way to our final and last cell fate, which is transformation. This is where the genome of the virus integrates itself into the genome of the host cell, which can either remain dormant and undetected, or its insertion can affect normal cell functions, depending on where in the host genome it is inserted. This also means that when the host cell divides during the natural cell division process, all progeny cells will contain a copy of the viral genome. And so if we were to visualize this, we can see that transformation can occur in both human and bacterial host cells, depending on the type of virus. And essentially, the virion will infect a susceptible host cell. And in this instance, we can see that the viral genome has been directly injected into the cell cytoplasm of a eukaryotic cell. It will then make its way to the nucleus, and using various viral proteins, it will become directly integrated into the host genome. Now, when this happens to a eukaryotic cell, the host cell is referred to as a transformed cell, indicating that it has foreign viral DNA within its genome. If this was to happen to a bacterial cell infected with a bacteriophage, it would become a prophage via the process of lysogeny. Now, in both instances, regardless of whether the host is a eukaryotic or a prokaryotic cell, the infected cell will at some point undergo cellular replication, and as part of this, the genetic material within the cell is duplicated. This means that both daughter cells will end up with a copy of the viral genome in their own genome, as it is copied indiscriminately during the replication process. Now, one of the biggest concerns for the transformation of human cells is that it can lead to something we call viral oncogenesis. And this is essentially where the virus puts the transformed host cell at greater risk of tumorigenesis, as the presence of the viral genome within the host genome affects its ability to perform normal cell functions. And there are two main ways in which viral oncogenesis can occur. The first is where the insertion of the viral genome increases the production of what we call oncogenes within the host cell. Now, oncogenes are genes associated with growth factors and hormone receptors. Therefore, if we were to have an unregulated increase in their production, cells could grow uncontrollably, resulting in the production of a tumour. Then on the flip side, insertion of the viral genome could decrease the production of critical genes such as tumour suppressor genes and those involved in the apoptosis pathway, which would essentially have the same effect, resulting in a cell that could grow uncontrollably. Now, when a virus directly interferes with the process of oncogenesis, resulting in tumour formation and increased risk of cancer, it is known as an oncovirus, and around 15 to 20% of all cancers are thought to be linked to these oncoviruses. So some examples are things like the hepatitis B virus, which is linked to liver cancer, the Epstein-Barr virus, which is linked to various lymphomas and carcinomas, the human papillomavirus, which is probably the most well known on the list and is associated with cervical cancer, and finally the human T-cell lymphotrophic virus, which is associated with T-cell leukemia. Now, it's important to note, infection with an oncovirus and the change in the growth of a cell does not always result in tumour formation or cancer. So as an example, here we can see the manifestation of genital warts as papillomas on the surface of the skin, and this is due to infection by the human papillomavirus and the cell transformation process. Now, genital warts are probably the most common sexually transmitted viral infection within the UK, and not all human papillomaviruses cause genital warts. Of the 100 or so variants that have been identified, only around 15 or so have been shown to cause genital warts, with 2 main variants being responsible for around 95% of all cases. Now, for the most part, infection with the human papillomavirus results in relatively benign growths, and this is the result of what we call low-risk strains. However, some strains are termed high risk, and they are associated with cancer, such as cervical cancer, making the human papillomavirus a potential oncovirus. However, it is dependent upon the infecting strain. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.